Spiro, thank you. That was a very kind and generous introduction. This is my first SID meeting, and I can tell you the last couple of days have been nothing short of just a treat, a real treat and pleasure. I was a little bit intimidated when I looked at this. It is humbling. It's humbling to be counted in the company of these stellar speakers, these stellar individuals, scientists. But beyond these award and honor talks, even the presentations that I've seen, the talks that I've heard from students, the posters, it's been invigorating. So thank you. Thank you for the invitation, especially Angela, the entire organizing committee, as well as Robin, who's been working a lot behind the scenes to make our lives easy. So thank you, everybody. It's a real pleasure to be here. I guess first things first, so I do have a few disclosures to note. Since you can read just as well I can read, I'm not going to go over them. I will note that I have no conflict of interest, either to this presentation or contents related to 3D printing. And all opinions and views in this presentation are entirely my own. And I would love to hear some disagreements, a spirited debate, perhaps over some adult beverages, maybe even some hippie juice later in the evening. But I should mention that I do have two other important disclosures. I'm an engineer. Even more damning, I'm a chemical engineer. So please don't hold that against me. Typically, as an engineer, our first instinct when we look at complex systems is to try and simplify them. So we'll remove some of this complexity, we'll try and get down to the simplest system, and then try to understand it. Now, of course, we don't always get it right. Sometimes we do some very stupid things, but luckily for us, swimming in our tank are other fishes like you, who understand the science, the basic sciences better, who are looking over our shoulders, giving us good advice, and stopping us from doing some of the stupid things that we might otherwise do. But I think there is a lot of value in kind of breaking systems down first into simpler forms, and then trying to take those systems, whether it be chemistry, whether it be biology, but then working back that complexity into those systems by developing the right kinds of platforms, the right kinds of approaches. And so the theme that you'll notice in today's talk, and somewhat even in my career as a student, and as an independent investigator, is I've always been interested in employing platforms for developing sufficiently representative models of very complex systems, and hopefully we can do something good with that. So here is the quick outline for the talk. I'm going to touch upon four aspects. I'll start with a little bit of an introduction to tissue engineering, if only to highlight the significant advances and how they've set the stage for new technologies like 3D printing to take advantage of what other pioneers have already done in tissue engineering. Of course, I'll give a brief introduction to 3D printing. I think we all by now know generally what the idea is. I'll share some data from our lab, but also allude to data from other folks and what they've been up to. And then, of course, as any scientist does, when you're thinking about technologies, when you're developing technologies, you want to do it so that people can benefit from it. So we have to think about the translational aspects of it. And I'll share my views on what the opportunities and challenges are for translational sciences, clinical translation. And then briefly touch upon social and ethical issues. I think in any kind of technology, when you're thinking about this bench side to bedside approach, you have to take all of these elements together and think about them as a whole piece. So hopefully this lecture will walk you through these different aspects. So let me start with tissue engineering. I'm sure this is a term that's well known to you. You have a general idea of what the field is. Nonetheless, it does have different connotations for different people. So one connotation is trying to take, and as this figure on the left shows, you can harvest cells from humans or animals. These could be stem cells, these could be differentiated cells, or differentiated cells that can be reprogrammed into stem cells, right? You can take these cells, harvest them, and then grow them outside of the body in an incubator, in a flask, add the right kinds of matrix components. They could be synthetic, they could be natural. And then if you combine them, if you have the right recipe for throwing all of these ingredients together and you wait just long enough, what you have is a miniaturized representation of that tissue or organ system. Simplistic, nonetheless, will cover some form, some function, help you ask important questions about how biology occurs in that tissue system. Right? So this is one connotation, and this has made a huge impact on drug discovery and drug development programs. Every day you hear about a new organ on a chip. There are many such organ systems that are now being represented in a massively miniaturized, massively parallelized way on chips. And these can be used to screen lots and lots of compounds quickly to get to phase 1, phase 2 studies. But I'm going to focus on a different context of tissue engineering, which is organ engineering. And I think it addresses a big scientific as well as a moral challenge that we have as scientists. So we all know organs are critical. I have some data here in the top right. This is from the Wall Street Journal in 2008, so it's a little bit dated. Nonetheless, what you'll notice is that the number of organs needed and the number of organs available from donors, there's a huge gap. And that gap just keeps on growing, and it's continued to grow as the average lifespan increases. But organs and tissues are lost to diseases during that increased lifespan. So how do we meet this gap? It has also caused a moral and ethical problem. So in developing nations and underdeveloped nations, poor folks are often lured by illicit practices into selling their organs, which are then traded in the black market at a premium. And so if we can really, as engineers, as scientists, come together in addressing how to grow organs outside of the body so that they can be brought back into the body for transplantation, I think we would have made a huge impact both on the quality and the quantity of human life. All right, so just to very quickly in one slide cover the key developments in tissue engineering over the last 4 decades. I will apologize, I can't cover everything in one slide. We all have to leave by Sunday, so I'll keep it short. Just the symbolic firsts, the key moments that have set the stage in advancing tissue engineering, but then also providing the right kind of stage for 3D printing technologies and other similar technologies. For me, this is the momentous event. Many of you might remember this iconic picture. It's from Brigham Hospital in Boston. This was the first successful kidney transplant between 2 identical twins. And I think it seeded the idea that an organ that has been growing outside of your own body, that has been conceived outside of your own body, can actually be brought back into your body, and it can recapitulate some of the functions of a diseased organ. And so after this, there were many firsts the next couple of decades brought together: liver transplant, heart transplant, pancreas transplant. And I think this really set the stage for what then seeded the idea of tissue engineering, that can we actually even make these organs outside of the lab, or in the lab outside of the human body. And I think the first, some of you might remember this, was this demonstration that a human ear can be grown in the back of a rodent. This is the Vacanti and Langer paper in 99 or 97. We'll come back to this a little later in the talk. But since then, in the next couple of, and it has also spawned some very popular themes in the media. Some of you might remember this reference too. Nonetheless, the next decade brought some advances where more complex organs could be engineered outside of the body in the lab. And so here are some examples of the bladder, the trachea, again from the Vacanti lab, vascularized intestine, the lung, this is from Meish's lab, and then even more complicated organs in the last decade, which include the liver, the heart, and an entire phase for transplantation. Some of you might get mad at me. Don't, I haven't forgotten. The first big development actually was engineering the skin. Now I have a suspicion this is a group that likes skin more than other organs. I don't know why, but it's just a guess. But this was the first time showing that 2 types of cells, fibroblasts, keratinocytes, could be taken together, embedded in a dermal matrix, and you would have something that would capture the function and the form of human skin. So in all honesty, if you think about tissue engineering, skin represents the first tissue or organ system that was successfully engineered outside of the body. All right. The one thing, maybe if I could go back, the one thing I didn't mention, which I should, is that there is still a key challenge. Most of these examples use an already existing animal organ that has been decellularized and then repopulated with the right kinds of cells. So if you think about it, you take the cells out, but you maintain the structure, and then you repopulate that structure with human cells. And this has really been the strategy that has worked the best. But the bottom-up approach of actually designing that scaffold, that support structure, that matrix to house the cells, that has been quite difficult. And there aren't that many great examples of entire organs where this kind of support structure has actually been engineered in the lab. And the way I see this is because of the kinds of complexities that exist in biological systems. Now, I don't want to be Captain Obvious. Everybody knows biology is complex, but I'm going to take a look at it from a slightly different perspective, right? From an engineer's perspective. Biology happens in 3 dimensions, but it also happens over multiple length scales. And what I mean by that is if you look at any organ, if you look at the basement membrane, if you look at the matrix, it contains many different proteins, growth factors, proteoglycans, hydrocarbons. So we are talking about the molecular length scale, right? So on the order of nanometers. These are arranged in thin membranes which can span a few hundred nanometers to a micron. Then you have cells which are on the order of 1 to 10 microns. So if you kind of take this impressionistic view of any tissue, I'm just taking one example here in the top left, you'll notice that many different components are organized over many different length scales. And that's what I mean, that we have to get all of these length scales from the nanometer level to the millimeter, centimeter level, the entire tissue level, right. Not only that, we have to get the precise placement in 3D. Like I said, biology happens in 3 dimensions. So we have to make sure that every single component of that organ is placed in the right context in the 3-dimensional space. I'll allude to this a little later in the talk. We have to have the perfusion and vasculature. Grafts can't survive on a host unless they can talk with the host. So there has to be the supply of nutrients, the removal of metabolites. And for that, you really need this supply system, this delivery system, communication channels. It's usually the vasculature that provides it. So we have to think about incorporating these features into the organs. And finally, something that I'm not going to talk about because a lot of other speakers have talked about this, but making sure that the immune system is on your side. When you're going to take this organ and then transplant it back into the host, there has to be an immune match, right? So I don't mean to suggest that we can't build complex structures. In fact, if you look at the human species, and there was a reference made to the book Sapiens, which is a great read, but if you look at our triumph as a species, as human civilization, it's largely based, if not completely based, largely based on our ability to design complex structures. And there's examples that you will find. This is a slight brain games. At the end of the talk, I'll ask you to remember all 11 of these structures and where they are. But we can build complex structures. But I will use these next couple of slides to draw the distinction why it has been so difficult to do it in the context of biological systems, right? And so I'm just picking an example of this beautiful building that I have the privilege to be in. This is the Center for Biotechnology and Interdisciplinary Studies on the RPI campus. And so if I think about, how was this building built? There are 3 key elements to it. One is you have the constitutive components, and so it's simple words, raw materials. You have your brick, your cement, your wood, metal, steel, glass, everything that goes into making this structure. Then you have the blueprint, which is essentially a code of instructions to guide you how to arrange these materials in the 3 dimensions, right? And then of course you need the construction equipment to pick these raw materials and then arrange them according to the blueprint, right? And so I don't mean to be pedantic here, but let's see if we can extend the same kind of philosophical approach to building biological organs, right? Here, the raw materials will be different. We'll have cells, we'll have matrix materials, proteins, growth factors, cytokines, whatever is necessary to feed the cells, to provide the right kind of communications across the cells, right? And we have technologies that have advanced that allow us to extract cells from tissues, from stem cells. We have scaffold molecules and growth factors that too can be extracted from tissues, or recombinant technology has come a long way that these can be made in other host systems outside of the human body. We of course need the blueprint, but we have very sophisticated imaging technologies now, and pick your favorite acronym, it's somewhere in there, but you can get whole body scans, whole organ scans, but then the good old classic immunobiology, immunofluorescence, histology can give you the resolution in 3D at even finer length scales. So you can now think of a blueprint that has that entire range from nanometers to the millimeters, centimeters being covered. But in tissue engineering, we've relied typically on the traditional tools of pipettes, glassware, and really good, capable hands of postdocs and graduate students. Now, I don't want the postdocs and graduate students in the room to be offended. I'm not calling you a tool. You've done some wonderful work, which over the years has helped us advance the field. But I hope you get the spirit that we don't have the tools. We don't have the tools to do that structural reorganization in 3 dimensions, right? And so that's really where 3D printing comes in. It fills that gap. Now, 3D printing is not new. The first patent on stereolithography was given out to Bell in 1984, I believe. And so over the years, this technology has developed. It has taken on many different names. By the time I'm done, there's probably a new name that's going to be coined. But essentially, and I'll stick with 3D printing for the sake of this talk, these names refer to the methods of deposition or the inks that are used. We'll talk about inks in a little bit. But these names refer essentially to how the printing is done. But what we'll do is we'll agree that no matter which one of these technologies it is and how the deposition is done, a general acceptable definition is that it's the automatic, precise, computer-aided layer-by-layer deposition of materials in 3 dimensions to generate complex structures in a rapid, reproducible manner and at varying length scales. Now, if we extend this definition to bioprinting, then we also have to throw in there that the structure that is being created has to be functional. It has to be compatible within the biological system. All right, so there are many different platforms. These first 4 on the screen, and I'm, again, differentiating them based on 3 aspects: the ink use, the fabrication mode, and then, of course, biocompatibility, because we want to extend these to build biological organs. And so the first 4, just to kind of give you a little bit of background, they either use a powder, a resin, or a polymer as the ink, right? And then used either heat or light-based curing of materials to get the form. Now, of course, these are physical triggers that are not always compatible with biological systems. So we rely on a 5th type of printing mechanism, which is designed to handle viscous gels, viscous materials that are gentle enough on the bio-ink, on the cells, that they wouldn't shear or die as they are being printed. And so there are three mechanisms shown here based on pneumatic systems, piston-based systems, or extrusion-based systems, right? And you can do line writing by applying pressure so that you're depositing the material at different locations the way you want to. Now, we use a slightly different version of that last set or last example of printers. Rather than do line writing, this does droplet printing. So in some ways, it's very much similar to the inkjet printer that you have on your desk. This is sort of the innards of the printer on that image on the left. But what we can do is we can do droplet printing at extremely high resolution. So we can go down to droplet volumes that are in the order of 10 to 100 nanoliters, and then spatial resolutions, by which I mean how far is one dot that's being printed from another dot. And we can get resolutions of 10 to 100 microns. Now, my colleagues in mechanical engineering who develop printers for completely different needs can get resolutions down to 10 or 100 nanometers. So we're getting down to that protein length scale that I was talking about before. The way the system works, and again, this is a simplification, it has a few cartridges. We have 4 cartridges because we ran out of money after 4 cartridges. But very much like the blue, green, black, and red ink in your desktop printer, we can fill these cartridges with different types of cells, matrix materials, it could be growth factors, right? And then we have a simple pneumatic system, a micro valve that pushes a column of air to dispense the material. Then we have droplets coming out. And then, of course, you have to print whatever university you are at to convince people that this thing works. And so we printed RPI with it and said it works. Anyway, so a more schematic representation would be you can print these combinations of droplets and matrix materials in a layer-by-layer fashion. So if you look at figure A at the very bottom, let's just say we are simply printing collagen, a layer of or a 2D plane of collagen. These are phase transitioning materials, so we can use salt or temperature to cause phase change. And then we allow that material to solidify so we can print a second layer on top of it. And we can do this in sequence by changing the matrix materials, changing the cells based on what type of organ system you're trying to produce. Now, skin is a great example because it already is very hierarchical, very layered, very compartmentalized. And so you will see many of these initial 3D bioprinting papers have used skin as a prototypical example because it's perfect for layer by layer assembly, right? Nonetheless, I'll talk about how we can go beyond just simply creating the epidermis and the dermis. All right, so the printer at the end of the day is a dumb robot. It needs instructions, and so your instructions look something like this. Print a layer, then print another layer, and then print these cells in that layer, and then print the other cells in another layer. And when you do that, at the end of it, what you get is a structure like this. Again, very simple. This is a reconstruction of 3D confocal images. And on the top, you'll see these nicely arranged keratinocytes in the epidermal layer. We are only staining for the nuclei, so you don't see cell-cell contacts. But we verified that we get good expression of tight junction markers. We get a very nice barrier function from the skin. In the bottom, you'll see the elongated fibroblasts. And again, they're not in animated suspension. There is collagen in that matrix. We're just not staining for it. But this gives you a sort of idea of what that printed structure looks like. And of course, we have to convince ourselves that if we do more detailed analysis and studies of these tissues using histology and immunofluorescence, we do see a very good match. I won't again bore you with the details of this figure. Trust me that we see very nice similarities between printed tissues and the traditionally created manually deposited tissues. At this point, you might be thinking, well, doesn't this seem like bringing a fire truck to put out a match? Why use some fancy stuff when all you're going to do is create something that I could just, the classic jam on the butter style, sheer collagen, and then put some keratinocytes on it. And if I get that model anyways, why do this? And people have been doing this for the last 50 years, right? In fact, there are clinical products out in the market. But again, like I said, they are somewhat reductionist, simplistic representations of what skin is physiologically. There are many elements missing from these simpler models. And really our goal is to try and see if we can start incorporating these, whether they are structural, whether they are compositional, right? And so the goal in our lab is, can we go from that very simple model on the left to something that's more complex, more physiologically relevant, but do so by employing the right kinds of platforms. And so this is where 3D printing plays in. It allows us to address those resolution, structural building gaps that I was alluding to earlier. So the first thing we decided to do was look at how much more complex can we make this simple skin matrix. So if you look at just the dermis, it has far more things in it than just collagen, right? There are proteoglycans, there are glycoproteins, there are other structural proteins like elastin, and all of these are necessary to reproduce the function of the skin, the mechanical properties of the skin, the tensile properties of the skin. And so we decided, why not think about combining these different components in a very systematic manner. So these could be different types of cells. And again, we're not including all the cells, right? Simplicity to then complexity. So we'll start with fibroblasts, keratinocytes, endothelial cells, and then pick a few matrix components, and then do a combinatorial study. So my background is in high throughput screening. I love high throughput screening. I love this combinatorial and rational design and screening of matrix molecules. So we've started combining these to create bioinks, right? Inks that are specifically designed to mimic or recreate a specific compartment or a component of the skin. And so you can imagine that we can have a ink specifically for the dermis, an ink specifically for the dermal epidermal junction, another kind of ink that's for vasculature, right? And so I will not show you the results. This is a little bit of a teaser. Shameless plug-in, if you may. One of my graduate students is presenting a poster on this on Saturday, and she has some really, really cool and exciting data. Cool and exciting to us. We'll see what you have to say about it, but I do encourage you to go see the poster. She's also presenting on Saturday at 2:06, abstract 542. So I will move on to the next part of the story, which is work from my other graduate student, Tanya. We've been interested in incorporating vasculature in the skin right now. There are not many or any skin models that have a robust vasculature being a part of them. So we have looked at different types of cells, their influence on creating vasculature. So we've looked at the type of fibroblasts, the source of those fibroblasts, media components, growth factors. And I don't have time to go into all of them, but you can see a very clear difference in the vasculature formed on that image on the left-hand side versus the one on the right-hand side. So we can control the branching patterns, the density of the capillaries, the diameter of the lumen, and incorporate this vasculature within the dermal bed. I'm going to quickly skip through the next few slides. This is showing you that indeed what we have is the formation of a vessel-like structure with a clearly defined lumen. And then we have the vasculature spanning the entire 3 dimensions. So it's not a planar vasculature, but it's indeed in 3 dimensions, innervating the upper dermis as well as the lower dermis. We're not the only ones. There are other examples, other folks doing this. Patterning of the vasculature is more common. And again, while it might not be physiologically representative, if you're thinking about skin on a chip-based systems where perfusion is necessary, examples like this could be quite important. In the future, we are hoping to tap into the power of stem cell technology, create autologous skin grafts, patient-specific skin grafts, start thinking about incorporating additional adnexal structures that are not part of the skin right now, sweat glands, hair follicles, sebaceous glands. And so that's generally the direction that we are going in. By we, I don't mean the royal we, but the we as a community. I want to quickly talk a little bit about what are the challenges we foresee. So right now, FDA has a 3-tiered classification system that's handled by CBER, CDER, CDRH. And typically, whatever you make for medical use is either a drug or a device, or it's some combination. There is this real nebulous space right now where we don't know what tissue-engineered 3D printed products are going to look like or organs are going to look like. What are they going to be classified as? And FDA doesn't know either. It's still a dialogue that is being had. So we have to think about what kind of regulatory processes would be defined for 3D print systems. What are the appropriate animal models? This is a question that's true for any kind of translational work. You have to think about what is the right physiological representative system. And some of these questions are similar to any kind of tissue-engineered materials, but there would be some unique, unprecedented questions specifically in relation to 3D printed organs. And then is the clinical trials framework going to look like it does for a classical drug? How do you get control groups? Typically, you need a few thousand patients. How do you get a few thousand patients where you remove a heart and put a bioprinted heart? What is the control group for that? So there are many, again, open questions that we don't know yet, and we will have to figure out to make all of these technologies become relevant to impacting human life. And of course, then we have to think about the public perception of all of this. If you remember that mouse on the ear, or mouse ear on the rodent work, it got a lot of backlash. This is an advertisement placed in New York Times in '99, where there are some very interesting excerpts. I won't go through all of them, but what caught my eye is, our society is not used to questioning technologies, but it's a skill we need to develop. So let's pose some questions, right? What kind of things do we see as being important, translating 3D printing of organs to patients? It could cause deepening of social stratification. These are expensive technologies. We've had an ongoing debate in the past few months. If you paid attention with the $67,000 MS antibody, the $37,000 eczema therapy, there are not that many people that can afford this. And so is it going to be cost equitable to everybody? What are the challenges within the untested paradigms of using living tissues, incorporating them into the body? We haven't done it before in much the same way. So there are, again, these open questions that we will have to think about. And then finally, the ownership of platforms, technologies, cells, tissues, and organs. This has become very quickly a rapidly growing area, and the landscape is a bit murky. Who owns the technology? Who owns the organs? There's a lot of other questions that need to be addressed. So finally, we are the ambassadors as scientists to tell the public about what is the newest and most exciting thing happening in science. And these things always spark a public imagination that can run amok. It has happened before, it's happening now, it will continue to happen, right? And so this is an article in The Economist that I saw about 3D printing, and I don't know what is going on here. Apparently, this is a surgery room where some organs are being printed on demand in real time. There's a heart, a liver, that might be a sausage for snack breaks during the surgery, I don't know. And then finally, this one really caught my attention is, will the next step be printing the entire human body? Now, this is where we draw the line. Engineers, scientists, everybody has to stop. Biology has given a perfectly good way to make human beings. It's far more fun than what 3D printing can do. Let's stick with that. And so, very quickly, I know I've run out of my time. I just want to acknowledge all the folks who've helped us, the vascular biologists, the dermatologists, all of my wonderful colleagues, without whom I would still be the fish breaking my own fish tank. I do want to acknowledge the 2 graduate students, Carol and Tanya, who've been working on this. And of course, people who give us money so that the lights stay on in the lab. And finally, thank you for your time and attention. This was a real pleasure. Thanks.